Hi, today we will be discussing about the isolation of glycogen. This experiment aims to explain the principles and processes of isolating glycogen. It also aims to determine the amount of glycogen isolated and the factors that can affect its yield. Lastly, it aims to enumerate some human disorders that can affect metabolism of glycogen in the body. Here we have the glycogen structure. As we've learned before, glycogen is composed of alpha-1,4 glycosidic bonds in its main chain. And compared to amylopectin, it has branches linked through alpha-1,6 glycosidic bonds. For the first procedure, obtain fresh clam or mussel and separate the flesh from the shell. Place mussel flesh in an ice bath. The reason why we need to keep the flesh in an ice bath is because its freshness must be maintained. Failure to put it in an ice bath may cause a lower glycogen yield in our results, as it will deplete in amounts if not kept fresh. Then, we need to homogenize the flesh through blending. 40 grams of the flesh will then be placed in an Erlenmeyer flask, which later will be mixed with hot KOH and heated in a water bath with constant stirring for complete digestion. However, from our experiment, it was observed that the use of a beaker is better than the Erlenmeyer flask, as it will give the experimenters more access to stir the flesh, thus better digestion. Digestion is the process of softening or disintegrating by means of heat. Gi-add na to ang hot KOH sa flask na nag-contain sa homogenizing of flesh, and then kana kay constantly giswirl na siya or gistir na siya for around an hour kay para ma-digest. So after the digestion, kay nag-result siya into a turbid solution. Ang question is, nganong hot KOH atong gigamit or nganong nigamit tag KOH jud? So this is because in high temperatures, KOH hydrolyzes proteins, lipids, and ribonucleic acids present in the tissues. With this hydrolyzation, glycogen can then be easily extracted. We know glycogen is a carbohydrate. This leads us to the question nga, nganong ang proteins, lipids, o ang ribonucleic acids naman ang ma-hydrolyze sa KOH, nganong di man niya ma-appeal ang glycogen? The reason ana kay tungod sa structure sa glycogen, di ba? Ang glycogen kay naay alpha-1,4 ug alpha-1,6 nga glycosidic linkages that are notably stable and can only be hydrolyzed by a strong acid or under acidic environment. Yeah, as we know, KOH baya kay base, so that means nga dili niya ma-hydrolyze ang glycogen. So in comparison, proteins, ngano ang proteins kay ma-hydrolyze man? Proteins have amide bonds which can easily be broken by either acidic or alkaline hydrolysis, while lipids kay naman sila ester linkages kay it can easily be hydrolyzed or broken down under alkaline conditions. The mixture was diluted with 15 ml water and transferred to a large flask, where 30 ml of 95% ethanol was added. The resulting mixture was then cooled and followed by centrifugation for around 5 minutes to collect the precipitates. The reason for the use of ethanol in this experiment is because it helps precipitate the isolated glycogen through removing the traces of water in the sample, thus leaving only the glycogen behind. The precipitate was then dissolved in 6 ml of cold 10% trichloroacetic acid or TCA. To remove the brown protein residue, centrifuge the mixture and retain the supernatant. Glycogen was recovered with the addition of ethanol and left to stand in an ice bath for 10 minutes. Here we have another reagent, TCA. The reason for the use of trichloroacetic acid is it helps increase the yield extraction of glycogen. How? Iyang iseparate ang habilin nga residue sa other macromolecules from the glycogen. This is then followed by the addition of water and reprecipitation through ethanol para ma-fully isolate and extract ang glycogen. The isolated glycogen was placed in a pre-weighed filter paper, let dry, and then weighed again. Weight of isolated glycogen is equals to the final weight of filter paper minus the initial weight of filter paper. Grams glycogen per 100 grams muscle is equal to grams glycogen over 40 grams of muscle flesh, which is the initial weight of the homogenized flesh, multiplied to 100 grams muscle flesh. Percent yield is equal to the actual yield over the theoretical yield multiplied by 100. However, there are certain factors that can affect glycogen yield. First, improper homogenization of muscle flesh can lead to failure of recovery of glycogen by the TCA. Second, inadequate swirling during saponification would lead to incomplete digestion. With the use of a Erlenmeyer flask, the swirling would be limited, and this would cause some of the proteins and lipids not to be properly hydrolyzed or broken down. It would lead to impure isolated glycogen. With the impure isolated glycogen, kay maka-add siya sa weight sa ato ang na-isolate kunuhay ng glycogen. So, simply saying, ang atong ma-isolate nga glycogen kay dili na siya purely glycogen, maapil na ang ubang mga macromolecules nga wala na hydrolyzed. So because wala na to gi-cover ang filter paper katong gi-dry na to ang isolated glycogen, na possibility nga ang presence sa contaminants kay kanang maka-add further pod sa weight sa isolated nga glycogen nato. Application of glycogen isolation. So we know that glycogen is the main source of energy for our body. It is stored in the liver, muscle cells. So when the body needs more energy, enzymes break glycogen down into glucose, which is then sent throughout our body. So because of this, glycogen isolation is needed for conducting researches regarding the availability of glycogen in different parts of the body to know and cure diseases or disorders that would affect the glycogen metabolism. There are various glycogen storage disorders that can affect the metabolism of glycogen in our body. First, kay ang glycogen storage disease type 1A or the von Gerd disease, which is caused by the deficiency of catalytic activity of the glucose 6-phosphatase because of the mutation in the G6PC gene. Because there is no glucose 6-phosphatase enzyme, imong body kay build up siya og mga glucose 6-phosphate. Why? Because your body can't form free glucose. The second glycogen storage disease is the Chorus disease. This is caused by AGL gene mutations that has a role in the making of the debranching enzyme. So we have discussed before nga ang glycogen para ma-use siya sa atong body kay kailangan siya i-break down into smaller units. So, di ba kanang alpha 1,4 man gihapon ng bonds nga nakakonect sa alpha 1,6 nga branch, ma-breaker na nila kung ma-activate na sila. So, ang mahibili na lang kay kanang limit dextran, which is alpha 1,4 nga main chain o connected sa isa ka alpha 1,6 nga bond. That's why kailangan nimo ang amylo 1,6 glucosidase enzyme para i-break ang alpha 1,6 nga bond gikan sa main branch nga alpha 1,4. However, sa kaning Chorus disease kay missing man ang enzyme, so ang mahitabo na lang kay ang mahabilin ra nimo kay kanang limit dextran niya, dili naman nimo ma-break into smaller units. So another glycogen storage disease kay ang Anderson disease. So this is caused by the mutations in the glycogen branching enzyme. So ang Chorus disease kay walay debranching enzyme, walang Anderson's disease kay wala siya branching enzyme. So, kung walay branching enzyme nga makamix sa alpha 1,6 bonds kay mahitabo na kay it's just a long chain of alpha 1,4 nga bonds niya. Supposed to be kay ang branching unta kay it is supposed to make glycogen non-toxic in your body. So, kay wala makana mahimo nga magpatong-patong ang daghan kay mga alpha 1,4 nga glycosidic bonds nga makakause og toxicity sa imong body. Thank you for watching and listening to my report.